Hello, my name is Neves and I'm currently a student at the University of Calgary, and I'm studying towards getting my undergraduate degree, which will be a major in biomedical science with a minor in anthropology. And this summer I'll be spending my time at the Hallgrimsson lab, which is located in the Foothills Hospital in Calgary, Alberta, Canada. The Hallgrimsson lab uses developmental genetics and 3D morphometrics, so we do a lot of imaging here. We have a couple of different scanners. We have this really big one that gets really good quality scans called an X-Radia. And then we have a couple of smaller scanners which we use for different things. One of them is an in vivo scanner and the other one's an ex vivo scanner. Here at the lab we have 5 undergraduates: Dima, Danica, Michelle, Sihan, and myself.
Hi!
We have 3 postdocs: Marta Marcini, Rebecca Green, and David Cates.
Hi everybody!
We have a master's student who's going to soon be a PhD student named Jay Devine.
No.
And we have a PhD candidate named David Aponte. And we also have a lab manager named Wei. I started at this lab 3 years ago in the summer of 2016, right after my first year, and then I was working on a project with Charles Roseman from the University of Illinois, where we were looking to build a genotype-phenotype map to understand what genes are involved in molar development. So that involved a lot of scanning of mouse mandibles. My role in that was to scan over 1,200 hemimandibles in one of our scanners that we have, and then to collect data on the surface area of each teeth digitally from these scans in a program called Amira. I presented that project a couple of times, and now we're on to testing candidate genes that we believe to be responsible for molar development, and these candidate genes came from a QTL analysis that was done with the quantitative data I collected a couple summers ago. And this summer we're continuing that kind of on the side to collect RNA from embryonic mice so that we can start to see if these genes are acting in ways we suspect them to be. So this summer, while I collect RNA for that project, my main focus will be a project that I'm working on with David Cates, where we will be looking at how growth hormone treatment timing, either early before puberty or late after puberty, affects mandible growth and development of growth hormone deficient mice. So for that, I have full cranial scans of these mice throughout different points in their ontogeny. And my role, what I've been doing so far, is to take these full cranial scans and to isolate just the hemimandibles. So basically I create a model of the mandibles, and then we use these models and we import them into a software called MeshLab, where I developed a landmark protocol, and I've been placing landmarks on different points consistently throughout each mandible. And what we do with these points is that we use them and treat them as 3D image data, which we then can import into different statistical softwares. We use R, and we use that to kind of analyze the parameters of each landmark so that we can begin to understand the different shapes and sizes within each mandible, so we can see what growth hormone treatment is doing to these individuals. So that's what I've been up to. I'm really excited and happy to be an SDB fellow. It's been a really great opportunity so far, and it's been really nice to meet everyone else. I'm excited to see what other people are up to in the SDB, and I'm excited for what's to come with the rest of the summer. See you all later. Bye.